TB. And Kiran will be speaking on DNA sensing in myeloid cells.
Woo!
Yeah.
Hi everyone, I just want to say thank you for being here. I know you're probably sacrificing prime nap hours right now. It's like 1 to 3, so very much appreciated. This will be over soon. So yeah, I'm going to talk about DNA sensing of myeloid cells and how that relates to tuberculosis. So just for some background, TB is the most deadly infectious agent worldwide, and on top of that is the leading cause of death for HIV-positive individuals. The current BCG vaccine has extremely poor efficacy, and as a live attenuated vaccine, meaning it contains a live but weakened bacteria, it cannot be administered to immunocompromised individuals such as HIV-positive people. So there's a current need, obviously, for a much safer vaccine, specifically a protein subunit vaccine to be administered to provide immunity across the world. So the most basic and safest vaccines basically contain only 2 components. First one being an antigen, which is a molecule that can be recognized by our own bodies to build immunity, and the second one being an adjuvant, which is something that stimulates the innate immune system, allowing to build immunity. My project will focus on these guys, which are adjuvants. So I work in the Stanley lab, and our lab has previously shown that these molecules called cyclic dinucleotides, I'll call them CDNs for brevity, are an effective adjuvant for both the current vaccine and for the protein subunit vaccine, and they they play in the STING pathway, which I'll walk you through. So when a bacteria infects a healthy cell, it'll inject its own DNA into the cytosol, which is where it shouldn't be. This DNA can bind to this molecule called C-gas, which releases this cyclic dinucleotide, hint, called CGAMP, and it binds to STING, which leads to the induction of pro-inflammatory cytokines. Cytokines are messenger molecules that help recruit very important fighter immune cells to the site of infection, so we want them. And that's how we can fight TB. On top of this, more interestingly, our lab has shown that when we can cherry pick, basically, immune cell recruitment of Th17 cells, or just a special type of immune cell, that leads to better outcome for the host. So we want the Th17 cell response. We want cytokines that induce the Th17 response. So for my research questions, we wanted to see whether CDNs could successfully induce cytokine production in vitro, and whether that could be predicted of in vivo results in a live animal. That's what in vivo means. And also, can we cherry pick certain CDNs that can elicit a Th17 response that can therefore be a suitable adjuvant for a potential vaccine? And like I said, for the experiment, we took a mouse, we harvested certain immune cells, macrophages and dendritic cells specifically, and transfected them with structurally unique cyclic dinucleotides, and then we did a bunch of fancy science stuff, and then we analyzed them via qPCR, another fancy science term, to see which cytokine induction happened between different cyclic dinucleotides. So we tested for cytokines that specifically are known to be important for eliciting a Th17 immune response. These are the ones here just for your reference. But basically, if we see high induction of these, that's good. That'll be a suitable adjuvant for our vaccine. So just look out for the next slides. So as in macrophages, we saw that specifically adjuvants V16 and V18 were the most potent stimulators of these cytokines. But then when we moved to dendritic cells, we saw something different. We saw adjuvant V19 being the most potent stimulator of these cytokines here. So in summary, the adjuvants seem to be acting differently in both of these different types of immune cells, with different winners per se in each different type of cell. And then we asked, okay, do we see the same patterns in human cells? So we looked at human macrophages, and unfortunately, kind of got pretty complicated, as naturally we're just humans. So there was no clear pattern that could be seen like we saw in the mice, but then again, we didn't look at dendritic cells, which will be very important moving forward, something to further research. For next steps, it'll be curious to see when we use human vaccine trials, which adjuvant produces the best efficacy, provides the best protection, along with the protein subunit vaccine for TB. Also, it seems like we don't fully understand the role of CDNs and their mechanisms in cells. That'll be further researched. Also, there needs to be more testing done on dendritic cells, like I mentioned before. So if you stuck with me that far, I'd like to give my thanks now. Professor Sarah Stanley for being crazy enough to hire me and give me a research position. I don't know why she thought that was a good idea. Robin Jong, who was my direct mentor, she answered all of my questioning and very neurotic person, so I was hard to work with. Of course, Professor Christine Madsen Barcelos, thank you so much for all your support this year. It was amazing. Google, I wouldn't be here without Google. I'm sure none of us would have gotten degrees without Google, so kudos. Also to the mice. Yes. Thank you.